Thank you so much. And I've had a fabulous day so far in talking with everybody, so I really want to thank all of you as well. So I just want to start by telling you a little bit of story about where I've been and then how I've gotten to where I am now. So my first foray into trying to understand this question is to begin to ask the question, are males and females actually different? And some of you heard today during lunch, I asked this question because I actually had a goof in my very first experiment in my graduate work. I actually performed all the surgeries in the females, and we ended up with exactly the opposite data from our hypothesis. We then performed this experiment in males, and we found out it actually conformed with our hypothesis. So it then began me to question, are males and females actually different? And so this is sort of the hormonal milieu that I think males live in. There's sort of an on-off button versus females, which are very much more nuanced. And I think that this is mainly why it is really incredibly difficult to understand where and why the sex differences actually occur. Men are typically utilized in all of basic science research. We always use male mice because they don't have a hormonal cycle, and because then, therefore, their hormones are much more consistent. Additionally, male cells are used. There's a whole reason why, even in pharmacological research, males are predominantly used. That allows us as women, there's much less data that's actually been acquired for us. And partially it's because we are so nuanced. And oftentimes I often think about the fact when people say there's no sex differences, they've actually included women in the study who are 18 to 85. Hormonally, we're widely different across that age spectrum. So I think it's really important to understand when we're looking at women, how we're looking at women, and what hormonal milieu for which we're actually looking at these women. So while I was at UT Southwestern, I was a basic scientist, and so I wanted to just sort of talk to you a little bit about some of the findings that I had while I was there. And I think these findings, they've been published for a bit, but it's probably sometimes fun to actually hear the investigator talk about the findings, number one. And number two, I think they're so incredible. I really want to see how this actually transforms into whether this is true for humans as well as for the models I'm going to tell you about. So we started looking, asking the questions, indeed, if there are differences between the sexes, let's focus on one of the two sex hormones, and let's focus on estrogens. And this is just a cartoon showing us that estrogens have a wide variety of different effects on a whole bunch of different organ systems. And so this is just sort of to say, okay, estrogens are obviously very, very critical, but in which organ system and how are they critical? It's then important to recognize that estrogens then bind to one of their many different receptors. And I think that the estrogen receptor field is getting even more complex. They bind to either estrogen receptor alpha, estrogen receptor beta, the GPR or the GPR30, as well as ERX. There's now different isoforms within the estrogen receptors. ER beta has multiple different isoforms. Estrogen receptor alpha has multiple different isoforms. And they also act as second messengers as well as transcription factors. So once you dive into the estrogen receptor field, it all of a sudden becomes much more complex than what it might appear just on the surface. But I was very naive at the beginning, and so I decided to say, okay, well, those of you also, you heard from my background, I was a nutritionist first. So I often think that I think from bedside to bench. So I wanted to first find a human model that actually was important, and then see if there was a mouse model that phenotypically characterized the human, and then I could delve back into the mice and mechanistically characterize what was going on. So in focusing on estrogen receptors, I had found this paper, which demonstrated that estrogen receptors are even important in men. And when you had this individual who was a mutant in the estrogen receptor, this individual ended up with obesity, insulin resistance, glucose intolerance, and cardiovascular disease. And as I indicated, there is also a mouse homolog, which is if you knock out the estrogen receptor from mice, this is a total body estrogen receptor, knockout mouse, you again end up with a mouse that's obese, is hyperphagic, has enlarged fat cells, the metabolic syndrome, and the like. So I then established then that estrogen receptor alpha must be very important, not only in humans, but also in mice. And so I decided to utilize this as my foray into the estrogen field. And the first series of experiments, and that's the one that I'm going to predominantly talk to you about, is focusing specifically at the role of estrogen receptors in the brain. Now, this is not to say my more recent work is focused on the role of estrogen receptors specifically in adipocytes. I've also now just recently published the role of estrogens on the beiging or the browning of adipose tissue and the effect on mitochondria within the brown adipose tissue. And if you're interested in any of that work, I'm more than happy to share that. But I wanted to keep this focused, so I'm going to rely mostly on our work that we did specifically at the level of the brain. And this was our hypothesis. That was that estrogens regulate energy homeostasis and metabolic function through the activation of the brain estrogen receptor. We went ahead and utilized mouse models to be able to delve into this. We utilized the ER-alpha floxed mouse. We bred it to the nest in Cree, and then we knocked out the estrogen receptor from the entire brain. We went ahead and phenotypically characterized this mouse, and this mouse looked very much like the whole total body estrogen receptor alpha knockout mouse. Now this, at first I thought, okay, well then the brain is the critical site of estrogen receptor activity and action, and that's also a naive focus. Yes, the brain estrogen receptors are very important with respect to body weight homeostasis, but they're not the end-all or be-all. The peripheral estrogen receptors also have a profound function. But to suffice it to say, when we did these experiments, the total brain knockout looked to have a profound effect with body weight homeostasis. We wanted to then ask, okay, that was nice for the whole brain, but which neuronal population, and we specifically focused on neurons initially, was the estrogen receptor critical for regulating food intake and body weight? So we went into a neuronal specific, we knocked it out of the POMC neurons, the SF1 neurons, and the like. And we characterized those mice as well. And we found, again, that estrogen receptors through different neuronal populations are very, very critical with regulating food intake and body weight. But that wasn't enough for me. I wanted to ask an even more important question, at least for me. Again, going back to the bedside, I wanted to say that's lovely. So we know that estrogen receptors are critical not only for males, but also for females in their brain. And at the same time, it was becoming very apparent that our diet actually has an impact on our overall body weight and health. So I asked the question, okay, if a male and female eat exactly the same diet, do you get the same metabolic effects? And believe it or not, that question hadn't necessarily ever been asked before. And so I wanted to ask the question again, if I give a male and a female the same food, do they have exactly the same biological responses? And so that's what I'm characterizing here. At the same time I was thinking about doing this, actually a little bit before that, one of my colleagues, Mike Schwartz, and his team had shown then that when you actually induce inflammation within the brain, this inflammation within the brain causes a whole host of diseases to include obesity. And this was all done in male rodents. So I wanted to know the answer. If I give the female the same diet, does she also get brain inflammation, and is that also associated with obesity and a whole host of metabolic consequences? So these are the objectives of our study. We wanted to ask the question, does the high-fat diet, or the obesogenic diet, induce inflammation within the hypothalamus of both the males and the females? And if so, is it only neuronal, or are there other cell types that are involved in this inflammatory event? And then what does the high-fat diet-induced inflammation, how does the high-fat diet-induced inflammation occur outside the hypothalamus? Is it only selective to the hypothalamus, or is it in other brain structures? And knowing that many of you have an interest in Alzheimer's and inflammation, I felt that this was very pertinent perhaps to some of the questions that you might be asking as well. So this is how we embarked upon our study. We went ahead and we utilized a Western diet, and it's important that I actually explain this diet to you because it was very human relevant. Again, as a nutritionist, I wanted to make sure that the diet that I provided my mice was something analogous to what a human might eat. There are 60 or 70% fat diets that people use in mouse models. I'm praying that no human is eating that level of fat in their diet, but I wanted to have one that was much more consistent with a human diet. So I chose this diet. So it's relatively high in fat, but it also is high in carbohydrates. So I think of this, or my analogy is it's getting a Big Mac and a Coke. So I give you that as a caveat. So we're going to look at inflammation now. Is it driven solely by the fat, or is it also driven by the carbohydrates? Love to engage that conversation with you, but my diet has both. All right, and this is just a composition of the diet, which will become really important in a moment. I will tell you that the diet is incredibly tasty. It tastes like cookie dough, and so whether I give it to my male mice, which is represented here, there's a significant increase in body weight. If I give it to my male postdocs, there's also a significant increase in body weight, and they love it. So this is often our afternoon snack is eating high-fat rodent chow. So one of the beautiful things about using rodents is that then you can sacrifice them at the end of your experiment. You can go in and interrogate their tissues. So this is the hypothalamus, and so we went in and took out the hypothalamus and measured just markers of inflammation. And just a few, there's a whole ton of them, but I'm just highlighting a few. So this is IL-1 beta, NLRP3, as well as IL-6. So consistent with what the Schwartz team had found, when you take a male mouse, he gains weight on a high-fat diet, his brain becomes inflamed, and there's a host of metabolic consequences. But the real question I had was what happens to the females? Now, those of you who do rodent research know that there's a problem with designing this experiment. Female rodents are always smaller than male rodents. So you either have to have weight-matched animals, now your females are geriatric compared to your males, or you have to figure out a way of equating your playing field. And I'm specifically asking the question about obesity-related events. So what I decided to do to sort of equate my playing field is that the males and females were given the exact diet, but I waited until they gained the exact same amount on the diet. So it's the delta. So you're looking at the difference between their initial body weight and then their weight on the high-fat diet. Now, you could design experiments all different ways, but suffice it to say, the females weigh a little bit less, but they have gained the same amount of weight on the high-fat diet. So this is not that they're eating less or anything. They're gaining the same amount of weight. That's a really important caveat with respect to how I ran these experiments. So that's just represented in the black bar. So now we carve out the hypothalamus from the males and females, and the female hypothalami are not inflamed relative to the males. And they're not even inflamed when you put them on the high-fat diet or the chow, even if you've noticed that the baseline is a little bit different. So you could say, that's just gene expression. We didn't stop there. We looked at the level of protein. The level of protein is significantly higher for this, as well as many other inflammatory markers in the male brain, but the female brain did not become inflamed. Again, she's gaining the same amount of weight. So she's eating the diet, she's gaining weight, but her brain is not becoming inflamed. So this is again looking at TNF-alpha, another protein marker, as well as we looked at the message. And so you might ask the question, okay, that's fine. Well, that's the hypothalamus, but some of you might be interested in other parts of the brain. So we looked at the hippocampus. Again, what we found was that the female brain did not become inflamed. The male brain became highly inflamed in the hippocampus. The cortex, I can tell you this is true for the liver and for the adipose tissue. So one might say, why would that be? And I've been asked that question a lot of times. But if you think about it teleologically, what are women designed to do? We are designed to gain weight in a healthy way. If we gained weight and we became highly inflamed, that would be probably really deleterious to reproductive function. So if you step back and think about what our main role in society has always been as women, child bearers, it makes sense that when we eat a diet, we would gain weight, which all of us in the room who are women know that we do gain weight quite nicely, but we also might do it in a very healthy way, in a very protected way. I often think that if men gained weight in a very healthy way, all of a sudden they would be the huntee instead of the hunter, because they would be so obese they wouldn't be able to go out and teleologically and be the hunters and gatherers. So there might be a teleological difference for why males, when presented with the same diet, gaining the same amount of weight, have a different response metabolically than do the females. It's just something to consider. We also were asked when we were doing this paper whether there was differences not only in the inflammatory response, but also in the anti-inflammatory markers. And so in this case, we looked at IL-10, and what we found is when the males were exposed to the high-fat diet, they had an increase in inflammatory markers, but also a reduction in anti-inflammatory markers, where the females did not. We also looked at beta-hydroxybutyrate. We had a discussion earlier today about ketone bodies. What we found is beta-hydroxybutyrate is thought to be anti-inflammatory. The female brain has higher amounts, and then on the high-fat diet, still higher amounts as well. So it's like the female brain is protecting itself from this inflammatory milieu. So one of the questions we asked to bring you back full circle, I was really interested in the estrogen receptor alpha. And this is just looking then at the arcuate nucleus, the third ventricle is there in the middle, and we're looking at the immunoreactivity for estrogen receptor alpha. And hopefully what you can observe is that in the male brain, there's a significant reduction in the expression of estrogen receptor alpha following the consumption of the high-fat diet. And you don't see the same level of reduction in the female. This is looking specifically then at the protein level. And again, you can profoundly see that there's a reduction in ER alpha in the male brain associated, not necessarily causative, but associated with that increased level of inflammation. So we wanted to delve a little bit further into this. So this is just sort of an interim summary. When fed exactly the same diet, gaining the exact same amount of weight, the high-fat diet causes a reduction in estrogen receptor alpha and an increase in inflammation in the males, but that does not occur in the females. So one question you might ask is, okay, well, maybe there's some other difference. So what we decided to do in this model is actually remove the ovaries, take away the estrogens, and see whether it's the estrogens or the obesity that's causing this change in ER alpha and the inflammation. So we removed the ovaries. When you remove the ovaries from a mouse, she gains a significant amount of weight, and there's a reduction in the expression of estrogen receptor alpha within the brain. This is looking at the hypothalamus. I could also show it to you in the cortex. And this reduction is again associated with an increase in markers of inflammation. So there appeared to us at this time to be this strong correlation or association with lower levels of estrogen receptor alpha, higher levels of inflammation. So the next thing we wanted to do was test the hypothesis that perhaps the activation of the estrogen receptor alpha protects against high-fat diet-induced inflammation. So in this series, we decided to go into cell culture and see if we could actually overexpress ER alpha to see if it actually protected the hypothalamic neurons from inflammation. But we had to establish our model. So what we did is we exposed the neurons to palmitic acid. Now, some of you might say, well, why did you do palmitic acid? Well, I showed you this sort of diagram of the fatty acid composition of our high-fat diet. And the highest fatty acid within our high-fat diet is indeed palmitic acid. It's also the most common fatty acid found in animals. It's accumulated in the hypothalamus of animals fed on a high-fat diet, and it induces the inflammasome. Now, we submitted this paper for publication, and the reviewers said, well, that's nice. But how do you know that palmitic acid is truly increased in the brain of the animals that are eating the diet that's enriched in palmitic acid? A reasonable question, because it would suggest, are we what we eat? Or is our brain tissue representative of the type of foods that we actually eat? So we went ahead and we actually explored this. And this I found to be one of the most profound pieces of data that we acquired. And it's profound in two ways. First of all, what you see is the male animal, when consuming a diet that's high in palmitic acid, notice there's a significant increase in the quantity or the amount of palmitic acid within his brain. Notice it's not statistically significant in the female. But I think even more striking, and I'll talk to you more about this in a few moments, is look at the differences in the baseline. So here you have two animals, a male and a female, on chow. Notice the male brain has way more palmitic acid than the female brain. And I was like, wow, does this mean that our brains, I don't even know whether human brains differ in the fatty acid composition that we're seeing that the mouse brains do. And this is even on chow. So imagine when you then, as I did here, expose it to a diet that's different, how does the brain process it, and does it do it differently by sex? So what we decided to do then is go back into the cell culture model. We used N43 cells, which will become important in a moment. We exposed those cells to palmitic acid, and then we measured markers of inflammation. And this is just looking at a time course. So we exposed again to palmitic acid, and we see a significant increase in TNF-alpha and IL-6. At the same time, we're seeing an increase in those two markers of inflammation. We also saw a reduction in the expression of estrogen receptor alpha. And this occurred again, not only at the level of the message, but also at the level of the protein. And it did not affect ER beta. So some might say, well, that's just all of the estrogen receptors are getting augmented. Indeed, this was specific for the estrogen receptor alpha. We then decided, okay, if that's true, if we go in and we overexpress estrogen receptor alpha in this cell culture model, we should be able to attenuate the level of inflammation, and that's exactly what we did in the last bar. I suppose I have a pointer. Yes, I do, over here. So you overexpress ER alpha, and you get a significant reduction in the markers of inflammation. So that was not necessarily good enough for the reviewers either. This was a paper that was really hard to get into publication, as you can probably tell. So the reviewers were brilliant because they said, aha, but that's neuronal culture. What does that represent? Is that a male cell? Is that a female cell? What is it? And so I was like, actually, they're right. I have no idea what the sex chromosome milieu of my N43 cells were. So I said, okay, then let's go ahead and do this in primary culture. So we went ahead, we sacrificed our animals, we took out the neurons from males or females, plated them this time in the same hormonal milieu, and again, we went back and probed to see what the response was. And again, what we find, and I find this, and this is one of those other aha moments of my data set, was that even after you take the male neurons out of the male body, plate them into a hormonal culture that is the same as the females, the male brains still responded differently, even though now they're in this plate of estrogens, when compared to the female brain. So does that say that it's due to the XY chromosome, or is that due to the hormones? I would say that these data might argue that it's due to the chromosomes probably and not the hormones, but really thought-provoking in many, many ways. Okay, so then we ask the question, okay, that's lovely, well, that's all neurons, but what about other cell types within the brain? And so this, I think, is slightly controversial now, but at the time, we asked the question, okay, microglia, what's happening with respect to the microglia? And in our model, we found that estrogen receptor alpha is not expressed in microglia. Now, some of you might have microglial cell cultures. We actually had purchased 2 different cell cultures of purely microglia, but we found one of them that had estrogen receptor alpha was contaminated by astrocytes. So in our pure microglial culture, and again, any of you who have data that differ from this, I would love to talk to you more, we find that there's no estrogen receptor alpha. And even more importantly, when we take our microglial culture, expose it to palmitic acid, and then estrogen to see if we can attenuate the inflammatory response, there's no effect of estrogen, which is consistent with the idea you need to have estrogen and estrogen receptor alpha in order to attenuate the inflammatory response. And again, I'm more than happy to discuss this more. So then we said, okay, perhaps the other cell type that might be critical then is the astrocytes. And so here what we did is we actually looked at just overall astrocyte levels between the males and females. And again, what I think you can appreciate is the males on a high-fat diet have astrogliosis, where the females, there's a slight increase in the number or the amount of astrocytes, but it doesn't achieve the same level that you actually see with respect to the males. And this is just looking at measuring GFAP as a marker of astrocytes. And again, a profound increase in the amount of astrocytes in the males that, again, we did not see in the females. And this is then just looking at, we did a primary culture of astrocytes, and again, what we saw was that these astrocytes actually contained estrogen receptor alpha. And so this is looking, and we did a primary culture of astrocytes, and again, exposed the primary culture to palmitic acid. And what we found, let's just jump through the data here, is that again, the male primary astrocytes in culture, in a female-like culture, when exposed to palmitic acid, again have a profound inflammatory response. So this is just a summary then. So we found that there is no estrogen receptor alpha in microglia. Again, I think this is controversial. I think it has to do with when you look, but again, love to have more conversations. We found that estrogen receptor alpha is definitely expressed in astrocytes. It has a profound anti-inflammatory role, especially when paired with estrogen. We also found that palmitic acid is inflammatory with respect to neurons. Estrogen receptor alpha is expressed within the neuronal population, has a very important role with respect to estrogens and estrogen receptor alpha with an anti-inflammatory response. And this is just looking at the relative contribution of the microglia, astrocytes, and neurons with respect to the palmitic acid-induced inflammatory response. Okay, so then we said, okay, that's nice. So now what we've done is we've actually now said that it's possible that this inflammatory effect is happening both at the level of the neuron as well as the astrocyte. But what mediates that response? All I've shown you is a correlation, right? Low ER alpha, inflammation. I haven't told you how it's actually occurring. And so this we actually collaborated a little bit with Bruce Spiegelman's lab when that they had determined that PGC1 alpha is a transcriptional regulator of estrogen receptor alpha. So we thought, okay, perhaps then maybe there's changes in PGC1 alpha that's changing the level of ER alpha that's causing the inflammation. And so we went ahead and explored this. And this is PGC1 alpha in the brain. There had been much less appreciated about the role of PGC1 alpha in the brain. It's all well known that it's very important in adipose tissue, but it's also very important with respect to brain. So the first thing we did was we acquired the mice from Bruce Spiegelman, which were PGC1 alpha knockout mice. We actually also did some co-localization with this PGC1 alpha antibody and found that indeed ER alpha is highly co-localized with PGC1 alpha positive neurons. We also found that our mice that lacked estrogen receptor alpha also lacked PGC1 alpha. and that the mice that lacked estrogen receptor alpha did not change PGC1 alpha levels, but the mice lacking PGC1 alpha had lower levels of ER alpha. Hopefully I didn't confuse you. So then we went ahead and we made some viruses to be able to manipulate both ER alpha as well as PGC1 alpha. What we found first, we also wanted to see, does PGC1 alpha just change when animals are exposed to a high-fat diet? And so we found here that if we expose males to the high-fat diet, there's a significant reduction in the expression of PGC1 alpha in the hypothalamus that occurs in the males but does not occur in the female. And remember, I also showed you there was also a reduction in ER alpha and an increase in inflammation. And so this is just also looking at the level of the protein. So then this is also looking at the other tissues, because you might say, oh, that's just the hypothalamus, but it's also happening at the level of the hippocampus, the cortex, as well as in the liver and the adipose tissue. So again, in all tissues, when a male is exposed to a high-fat diet, PGC1 alpha goes down and ER alpha goes down. There seems to be an association between the two. And so we also asked the question then, does that happen even in primary neurons? And again, I'm always struck by this. Every time we take the male neurons out and plate them, even though they're in that estrogen milieu, we're still seeing the same profound effect on reducing PGC1 alpha that we saw in the animals. And this is just also looking then at the N43 cells. So if we take the N43 cells, expose them to palmitic acid, we can reduce the level of the expression of PGC1 alpha. And so the question here is, and it's relatively a complex study, is then if you overexpress ER alpha, even though PGC1 alpha is low, can you attenuate the inflammation? And indeed you can. So this is showing us, it's like the cart versus the horse. We think PGC1 alpha is the upstream mediator of the inflammatory response. So this is all well and good, and we were able to then show that there is a strong association between inflammation and that we're seeing following the consumption of the brain. But again, I'm this bedside person, so it's like, okay, that's nice, but what does that have to do with whole animal physiology? And there had been several papers that have been published that demonstrated that when you have an increase in brain inflammation, this is associated with the metabolic syndrome, and specifically changes in cardiovascular disease. So we thought, okay, so we have now a mouse model where animals are gaining the same amount of weight. We're seeing inflammation in the male brain, but we're not seeing it in the female brain. What happens on some sort of secondary output, such as cardiovascular disease? And so this is just sort of the hypothesis then, and that is that brain inflammation is associated with cardiovascular disease. So we paired with some cardiologists at UT Southwesterns, and they actually ran echocardiography for us. And what they were able to demonstrate is this fractional shortening is a determinant of cardiovascular disease. And this is sort of the actual raw data, but when we graphed it, what we found was those animals that had an increase in brain inflammation had fractional shortening, which was associated with decrements in overall myocardial function. So this again says the brain is important. When you have brain inflammation, it actually can cause things such as changes in echocardiography and changes in fractional shortening and cardiovascular disease. And again, the females were protected from that. So this is just a summary of all these data, and that is that when exposed to a high-fat diet, gaining the same amount of weight, you see this strong sexual dimorphism. The males have a reduction in PGC1 alpha, a reduction in ER alpha, profound inflammation, and thus led to myocardial dysfunction, and the females were completely protected. So then I asked the question, okay, so based on those data looking at the palmitic acid content, do males and females actually process the fatty acids differently? And again, I'd never seen this question asked before. So we looked not only at the fatty acids, but we also looked at ceramides and sphingolipids. And again, the male brain is different with respect to its expression of different ceramides as well as sphingomyelins and sphingolipids, such that the male brain with the inflammation has higher levels of ceramides, glucoceramides, and sphingomyelins when compared to the female brain. And there's a strong link between these and inflammation and cardiovascular disease, as well as Alzheimer's disease as well. And so this is just to show us, too, that when we actually add palmitic acid to a cell culture model, we see this increase in ceramides. But if we also treat the cell culture model either with estrogens or with estrogen receptor alpha, it attenuates the level of the ceramides. So again, there seems to be an important link between estrogens, estrogen receptors, and these ceramides or sphingomyelins. We went ahead and did an entire lipidomics analysis, and again we found that based on this, even lipid species differ between the males and females in their brain. And this was really, really amazing to me. So it says that when a male is eating a diet, and as a dietitian, I would always give a male and a female the same diet. It might differ in in the total calorie content, but it was always the same macronutrient composition. But perhaps that's a disservice. If females process nutrients differently, maybe females can have a little bit more fatty acids than males within their diet. But again, this has not been proven in humans yet. One of the fatty acids that I found to be most strikingly different, we did this in collaboration with Vanderbilt, was linoleic acid. So we actually sent the brains of our animals to Vanderbilt, and they said, oh, you must have had a problem. The male brains have absolutely no linoleic acid in them following the consumption of the high-fat diet. We said, well, that can't possibly be true. We went on and did additional lipidomics analysis, and again, not necessarily negative like this, but we always see a significant reduction in the amount of linoleic acid in the male brain following consumption of the high-fat diet. And why is this important? Well, those of you who are in the Alzheimer's field know that linoleic is essential for brain development and growth. It's also, its deficiency has been identified as a marker of early phase neurodegenerative disease. So this week we thought that's really interesting. What if the fatty acid milieu of the brain actually is driving some of these neurodegenerative diseases? And what if then in the male we were able to give them a diet that was enhanced in linoleic acid? Would that reverse some of the deleterious effects we'd actually been seeing? So we sort of did that in cell culture. We went ahead and gave it the cell culture either palmitic acid or linoleic acid, and when we gave the linoleic acid, it attenuated some of the markers of inflammation. It also restored the level of estrogen receptor alpha. I don't know the connection between the fatty acid and the restoration of ER alpha, but it also restored the level of PGC1 alpha. Again, I don't necessarily know that correlation. So clearly we found that men and women were different, and I was very, very excited about all of these findings, and I really wanted to know, that's lovely for a mouse model, but what about for the human condition? So this is when I transitioned and moved to Cedars-Sinai, and I decided to go ahead and embark upon translational research. But being somewhat, again, naive, I wasn't exactly sure how I was going to embark on translational research. So there was a lab that was actually taking these pluripotent stem cells and transforming them into hypothalamic neurons. I thought, that's the perfect way for me to do translational science. I can take human pluripotent stem cells, transform them into neurons, and see if these human cells behave the same to perturbations such as I had already explored with my mouse model. And I was even more lucky than that, which was they had pluripotent stem cells from obese men, lean men, obese women, and lean women. And I thought, this is the payday. This is exactly what I want to find. So I went ahead and interrogated some of my critical model or critical sites. I looked at estrogen receptor alpha, IL-6, and TNF alpha. And I found that the data could not have been, I'm sorry, I went too fast, could not have been more opposite from my mouse model. There was absolutely no correlation, no association with what I had seen in my mouse model with these human pluripotent stem cells. So I had to step back and I asked the question, why are these data, these preclinical observations so different from what I had seen? Is it because humans aren't mice? Now, all of us who do mouse research, we pray that's not the case of why the data were not transferable, so I'm going to bypass that one. The next is, what other issues could account for these differences? And is there something that happens during the culturing of cells that could actually augment these results? And it was right there that I became very, very concerned about what I was doing. And the question is, does the cell culture media actually influence my results? So I'm going to now transition into where we've been sort of stuck. And that is just to remind us that oftentimes when we see cell culture data, the cell sex is underrepresented or underreported. Most of our transformed cell lines actually probably have XXY. The sex chromosome is not the way we would anticipate it to be. And oftentimes, therefore, we don't even know what it is, so therefore it's underreported in many of the transformed cell lines. And that becomes a huge disservice because we don't necessarily know what we're actually dealing with. The other problem is that when you actually take a cell out of a human and you put it into a dish, the cell culture media does not replicate physiological conditions. The serum is widely variable with batch-to-batch inconsistencies. Many of you have had that happen to you. You buy a lot of serum, you have these phenomenal findings, you go back to replicate yourself, and you can no longer replicate yourself because you have a new batch of serum, and you have no idea what that serum constitutes because it's ill-defined. The glucose concentrations used in cell culture actually also exceed those found in vivo. The micronutrients used in cell culture differ from in vivo. The electrolyte concentrations differ, but even more to my point, the sex hormones differ. Specifically, when you take these pluripotent stem cells and you transform them into hypothalamic neurons, you have to use a growth media that's enriched in estrogens in order to make that transformation. That transformation reduced the level of the expression of the very estrogen receptor that I have spent my entire life looking over. So this became not necessarily a model that I actually could use. So not only are the estrogens that you use are in the cell culture media, but there's also phenol red that represents a non-steroidal estrogen, and even the plasticware is filled with polystyrene, which also imparts some level of estrogens. And so the serum, again, contains these estrogens. So I was telling some people earlier today, so I walked away from the lab after realizing all of this, and I was like, what a mess. And I will remind you that I live in Beverly Hills, and Hollywood happens to be where the Jenners have their favorite restaurant. And I was walking home, and there was Caitlyn Jenner. And I thought about my cell culture model, my male cells that are now in a milieu of female hormones, and I looked at Caitlyn Jenner and I said, that's what I have. I have Caitlyn Jenner. So let me just explain to you what I'm talking about. And that is that for the new terminology is cis male and cis female. So a cis female is a female who has 2 X chromosomes, but her X chromosomes live in an environment where estrogens are higher than testosterones. A cis male, on the other hand, is an individual with an XY chromosome who lives in a hormonal milieu where testosterone is greater than estrogens. We can now all appreciate, although we're not necessarily appreciating it, that sex and gender are not the same. There is a subset of individuals where sex and gender do not have a Venn diagram overlap. And it's those individuals that I think become really, really interesting for us to be able to explore more. And we have to remember then that gender identity is across a whole continuum. And we very rarely even consider gender in any of the biological experiments. You can't do it in a rodent model, but in our human condition, I think this is an area we need to explore in much greater detail. But if you think more about my experiment when I took these human pluripotent stem cells, especially from my males, plated them in estrogens, what I ended up with was a transgender female, where now I have an XY chromosome that's in a milieu where estrogens are higher than testosterone. And I have in the transgender men, it's an X chromosome where testosterone is higher than estrogens. I bring this up because I think it's really important as we start to interpret some of the cell culture data, what we're actually doing. And I think eventually we're going to be able to describe, is there an optimal ratio? And I looked at some data today with respect to the androgens and the estrogens. We oftentimes just measure estrogen or androgen and we claim there's a disease associated with it, but it may not be one or the other. It may be the ratio of the two within a sex hormone, a sex chromosome complement that actually confers disease risk. And this is an area that I think that is much underappreciated and it deserves some study. And so this is just sort of, as we're looking towards, it always makes me smile, we're looking towards personalized medicine, but we haven't even personalized medicine based on sex. So I think we need to step back a bit, look at how we can actually incorporate the sex chromosome and sex hormones and trying to define personalized medicine that way. I was just asked to give a talk actually at a transplant meeting, and this was really amazing. Many transplants, whether it's kidney or whatever it happens to be, they haven't been paying attention to the sex of the organ that they're transplanting into the individual. So I think of this as transplant, where you're actually, again, taking a female organ and putting it in an XX female organ and putting it into a male who now has a testosterone complement that's larger than estrogens. Is that not a transplant experiment? And what's happening with that tissue within that milieu? And it was really interesting. Evidently, ethnically, there are societies where only women donate organs, so most of the men receive female transplants. Again, this is an area that's probably ripe for exploration about what happens with those organs in that unusual hormonal milieu. So then just to end here, I think that cell culture needs to factor in cell sex when we're culturing these conditions in our overall analysis. Health is influenced not only by sex, but also by gender. And how are we going to start factoring gender? How do we even assess gender? And what does its influence on overall health mean? And what is the role of sex and gender when we're exposed to transplant or any of the types of experiments that we're doing? So with that, I'd like to thank you for your attention and thank all the people that assisted me in this work and take your questions. So thank you. Yeah. I don't know. Oh, I think there's a microphone.
Thank you, very nice talk.
Thank you.
So my question would be about age. I mean, we know that with this increasing age, we have maybe lesser of estrogen. So how would you see that? Do you think that the inflammatory markers would increase or decrease? And have you tested this post-mortem brain tissue?
Fantastic question. My belief would be that as estrogens decline, there would be an increase in inflammation because I believe that the estrogens are facilitating the protective effect. The closest thing I was able to do in my rodent model was remove the estrogens and see that we also got the inflammatory effect, but I think age is critical. And I think now that NIH has now mandated not only sex be included as a biological variable, but age, I think we're going to open up a whole new series of experiments that are so critical with respect to age. Yeah, I think age is a huge factor, but all of my studies were done with age being a constant and not with longevity being a factor in the analysis. Thank you. This question? Oh.
Thank you. Wonderful talk. I have a question along the same lines as previously. So in your overoptimized models, did you try to replete with estrogen? Is there a window in which repletion is successful? Or is there a window of no return for the estrogen receptors?
Great question. So what we find is if we instantly replace, so the animal, you remove their ovaries and then instantly put in an estrogen pellet, you attenuate that weight gain. So it's much more beneficial. If you go in and provide estrogens after they've gained weight, you get a much smaller effect of the overall estrogens. I think of it analogous, we were talking about sort of the women's health trials where you either provide estrogen during the perimenopausal phase, the timing hypothesis, where it appears to be most efficacious. But those women who've gone a long time without estrogen, now you give them estrogen, now it becomes deleterious. So I think that there is a timing hypothesis that we see in the rodents as well.
Deborah, that was great. I was curious with the high-fat diet study. Do you think, could you speculate on whether if you pushed the fat content even further, if you think there would be inflammation in the brain and in women? Do you think it's a threshold thing, or is it just really strong protective effects that whatever the poor diet looks like, that women are protected against?
We did try the 60%, and we still saw a level of protection. And I think that many people who do sex differences, and maybe there's even people in the audience, who, if you put a female on a high-fat diet, you're usually annoyed by her because she doesn't get insulin resistance and she doesn't really gain weight very quickly. And that's one of the reasons females haven't been used in some of these metabolic studies is because they lag all of the metabolic sequelae. So even at 60%, she didn't have as great of an inflammatory response as the male. We even did this short term. So these experiments are run over several months. We also did like 3 days of exposure, thinking if we hit them with a rapid increase in fatty acids, would you see this profound inflammation that then would attenuate? We still didn't see it in the females, but the males had a profound effect. So again, as I think more and more about my data, I think it could be a protective effect, but that's, I'm not sure.
Just to follow up, were those adults, and do you think there could be like a critical window when women might be not as protected if it was earlier in life?
I couldn't agree more. I think that there is probably I bet that the pubertal transition is critical for women, but I wasn't able to explore that in these models. However, The time for which we take the neurons out or the astrocytes out, those are more of a neonatal animal. So those data would say we're protected even early on in that model because you still didn't see that robust effect in inflammation to the palmitic acid. So it might suggest that we're protected earlier on, maybe.
Yeah, very nice talk. In your in-dish neuronal experiments, of course, there were no glial cells around, right? Would it be of value maybe to explore what male cells are like in a female glial environment and the other way around? Could that be interesting to test?
That would be amazing, right? To take to expose the male cells to the supernatant of the female cells, or to do sort of I think that would be amazing, right? So the milieu of the male cells was just identical to the females, but it would be interesting to take the hormonal milieu of a female and expose those male astrocytes or neurons to that more natural hormonal milieu. That would be amazing. Because I don't know at the end of the day, would you have a reduction in the inflammatory response? It's a great question, a really great question. I didn't do that. Would love to.
Hi, that was a great talk.
Oh, thank you.
So I keep thinking about estrogen receptor expression in humans versus rats. And as far as I understand it, there's a different role of, obviously, androgens are converted into estrogens for sexual differentiation in rats. We don't know that the same thing is true in humans. It looks like it might not be. And I'm wondering, first of all, what you think about how crucial that prenatal period is in terms of sort of setting the course for estrogen receptor expression in adulthood, and what you think about how your research on rodents and estrogen receptor expression translates to humans, given we don't, as far as I know, don't really understand the significance or locations of estrogen receptor versus androgen receptor, say,
In humans.
in humans.
Right. No, I think it's a question that I'm dying to know, right? Do we see the same level of expression within the same neuronal populations, within the same nuclei within the brain that we see in the rodent? Is there a mirror with respect to the human? And then is there a sexual dimorphism with respect to the expression of this estrogen receptor that we see in the rodent? Do we also see that in the human? Really great questions. And in the rodent, in the hippocampus, you can actually make estrogens, so there's actual local production. Forgive me, I don't know if we know that about humans. Do humans' hippocampal neurons also make estrogens? I don't know, but that could be another difference where you're having local production of a hormone, where you're taking either androgens and or estrogens and then looking at the relative ratio in those areas between the estrogen receptor and the androgen receptor. Phenomenal. Needs to be done. Yep, don't know.
Thank you. Can you hear me?
Yeah.
Thank you. So along these lines, about regional specificity of the sort of resting state, inflammatory condition within the hypothalamus in particular, about regional specificity and the responsivity, is that something that can be delineated?
Right, it's a good question. So because we went to other areas in the brain, we saw the same inflammatory response. So it didn't necessarily look like it was regionalized. Is that the way to answer your question? Maybe to just the hypothalamus. I think you're also seeing it in other brain regions. But the question is that true in humans? Is the big question, right? I don't know the answer to that. The idea is that the hippocampus is relatively leaky, and so whatever's coming from the periphery, the hippocampus might be much more sensitized or sensitive to whatever's coming up from the periphery, and so maybe that's where it really makes a difference, but I don't know that for human per se.
And do you know have you looked at stress and whether it modifies any of these inflammatory responses?
That's a great question as well. My animals, I don't think were stressed. We talked a little bit about, you went there at lunch, where I've had the benefit always of having a female room and a male room for all of my rodent studies. That's a blessing because then the pheromones are not circulating and then the males are not stressed by sensing female pheromones. So I probably was in a stress-free environment, but I don't know the impact the stress would have had otherwise. I guess I'm anthropological. Anyway, yeah. So, yeah.
So thank you. Really, really interesting. Going back to the resistance of females to the high-fat diet, have you looked at other things going on in the gut, specifically the gut microbiota? So one of the things that we were able to show is if you disrupt the gut microbiota with antibiotics, you can induce really a lot of gain in fat and metabolic dysregulation in females. So have you thought about that?
No, we were just talking about it just a minute ago. I think that's a really, wouldn't it be amazing to do a fecal transplant of male gut microbiota into a female and see if now she responds to the high-fat diet with an inflammatory response? Love to do that study. We didn't do it, but I think it needs to be done because as people are doing these fecal transplant studies, what if it's something as simple as that? You know, what if we gave all men our fecal biota? Right? But I don't honestly know. But I think it's a really great question. I mean, we were talking. I've seen a paper in a grant on PCOS that you can do a fecal transplant with a non-PCOS woman that ameliorates some of her PCOS symptoms. So they're definitely, so that suggests to me that those sex hormones are having an impact on the biota, and then maybe you could induce that or change that in a beneficial way. Be very cool.
Thank you for a great talk.
Thank
I
you.
just wondered, you focused your research on ER alpha, which makes sense, but I just wondered if you've looked at ER beta, either through maybe ER alpha or beta selective agonists in your mice, or maybe through in your cell culture.
Thank you. I appreciate that question. We haven't been blind to ER beta. I've sort of resisted it. We always look at it, but I don't feel like I look at it in a good way. The antibody for ER beta is notoriously not great. And there are like now, I think 10 or 15 isoforms of ER beta. So I always worry when I'm just saying ER beta doesn't change. I think I'm being grossly obtuse. I think that it probably changes in ways that are really important. And just like it's the ratio of estrogens to androgens, I think it's the ratio of the two receptors. And I think if you threw GPR30 or GPR in there, it probably has a role as well. We've made triple knockouts who are just, massively obese, and we've looked at trying to play with it, but I think we're missing some of the nuances because of the antibody issues. But I think that's really important. Yeah. Other questions?
So, great talk. With the females, you made the good point that they always gain less weight than the males. So what happened in terms of their total food intake on the high-fat diet? Did they eat as much as the males, so that's my first question. And then, what do you think would happen if you used a cafeteria diet as opposed to just the high-fat diet?
So I'm going to answer your second question first. That would be a great question. Again, that's why I kind of harpened on the fact that mine is at least high in carbohydrates and also in fat. But if you did a cafeteria diet where you had cookies and other things, would you see the females eat more of it and gain more weight? And would that become inflammatory? I don't know the answer to that. It would be an interesting question to ask. With respect to your first question, they didn't eat as many calories, but calories per gram, I think it ended up being of body weight. They ended up eating something closer to the males, but as total gram of weight, they didn't eat as many grams, but as a calorie per gram basis, they were closer to the males than the other. But the cafeteria diet would be very, very interesting to see what happened with that, for sure. Yeah. So one of the other ideas, too, would be if you remove the ovaries and then you put them on a cafeteria diet, do you accentuate the obesity that's induced by the ovariectomy? And we've done that experiment, and they get much, much larger. So it's the combinatorial effect of lacking estrogens and the high-fat diet really exacerbates the obesity.
I speak up. In relative amounts, the estrogen receptor alpha, did it decrease more in neurons or in astrocytes?
Great question. More in neurons or astrocytes? I know that we know those data, and I don't know it off the top of my head. But it is a really good question because we were really, which is the cell type that's driving this, right? I'm very, and I think sort of analogous even back to the question back there, which would be, how about co-culturing? What if I took a male neuron and co-cultured it with a female or with a male neuron and a female astrocyte? What's that communication, and what would we find that way as well? But I know we have the data about the relative amount, but I think that's the co-culturing, or even bringing all three together, the microglia, the astrocytes, and the neurons in one culture, a triculture, would be interesting as well. Yeah, for sure.
Well, thank you very
Thank you.
much, Deborah, for joining us.
Thank you.
Really appreciate it.
I just want to say that I wish I was all of you, because the list of speakers you have after me is totally phenomenal. I want to come back just to hear them. So please, you have an amazing slate. So enjoy.
And I also want to thank Laura Halson for inviting Deborah.
Thank you.
Thank you.